Environmental DNA is the DNA collected from the environment, that is, from environmental samples, such as, for example, seawater. The seawater contains small organisms, such as phytoplankton and bacteria, but it can also contain traces of larger organisms, such as tissues and cells, released by larger organisms, such as fish. By filtering a few liters of water, we can collect those traces to sequence their DNA and then analyze which species inhabit that environment. There are three main steps in the process of environmental DNA analysis: collecting water in the field, obtaining DNA sequences in the laboratory, and bioinformatic analysis of those sequences to see which species they correspond to. In some cases, we may also be interested in seeing if a particular species is found in a certain area, for which we use a technique called qPCR. For the first phase, for sampling in the marine environment, we usually take advantage of oceanographic campaigns, which allow to collect and filter water in the open sea at various depths and covering a very wide area. The second phase is usually done in the laboratory and consists on extracting the DNA, selecting that DNA of the species of interest, for example only fish, using specific markers, and sequencing the resulting products. The sequences obtained go through a series of bioinformatic data flows that are finally compared with a reference database to obtain the species to which they correspond. With the analysis of environmental DNA, we can monitor the diversity present in a water mass from seas and rivers, including areas of difficult access, without damaging ecosystems or sampling the organisms. For example, using environmental DNA, we can detect whether migratory species, those that spend part of their lives in the sea and part in rivers, are able to cross certain obstacles in rivers on their upstream migration, and without having to use invasive methods such as electrofishing or traps. We can also study the fish community present in estuaries to determine their environmental status without having to fish them and thus disturbing the ecosystem. But returning to the marine environment, during the Bioman scientific survey, we compared the species obtained through pelagic fishing and the analysis of environmental DNA of water samples. We detected about 16 species with the pelagic fishing and about 120 with the environmental DNA. In addition, we found that for species common to both methods, there is a correlation between biomass, kilograms of each species, and the number of DNA sequences, suggesting that environmental DNA can also be used to measure abundance. We have found that environmental DNA is distributed in vertical layers, which is very important for the study of the deep ocean. By analyzing the DNA of water from various depths, from the surface to more than 1,000-meter depth, we see that the DNA is stratified and that this stratification is also different during the day and during the night. This means that the DNA of some species is present in shallower layers at night and in deeper layers during the day, which coincides perfectly with the known vertical migration movements of some species in the mesopelagic zone and gives us information about the spatial and temporal distribution of these species. Environmental DNA has many advantages over other methodologies for exploring aquatic environments. It is not invasive, it does not damage the environment and does not require organismal sampling. It is objective because it does not rely on taxonomic expertise for species identification. It is accurate because it can detect the species that are not caught in nets, are not very abundant or are elusive. It also allows to explore inaccessible environments in a simple way. All these advantages make environmental DNA analysis very promising for cost-effective monitoring in the marine environment. But every innovative method comes with its challenges and opportunities for improvement. For example, it must be taken into account that finding DNA from one species does not always mean that this species has been there, because we may have contamination sources or currents that are moving those DNA from different places to others, or the DNA could be present in the stomach of a predator, for example. Therefore, it is necessary to interpret these results within their context. Something very important for environmental DNA analysis are the reference databases, because if the sequence of a species is not documented in a reference database, we will not detect it, even if it's there in our sample. Therefore, many developments are currently underway to improve and standardize the monitoring of the marine environment using environmental DNA. It is anticipated that in the coming years, this method will be routinely applied to replace some or to complement other monitoring campaigns.